Okay, so today's lecture is going to be on protein synthesis, and basically what that means is how do we get the information from our DNA code into all of the traits that we see and express on a daily basis. So we have to talk about going from genes to proteins. Here's the list of objectives that you should be able to accomplish after watching this OV. Make sure that you read them over, and when you get to the end of the OV, come back and try and answer those questions with multi-sentence answers. Okay, so first let's do a quick review of what we already know. From the landmark experiments with Watson and Crick, Mendel's contributions as well, and everyone in between, we know that genes are made of DNA, okay, so they're nucleotide sequences, and the proteins come from our sequence of genes. Therefore, the DNA must control the production of proteins, so the two are linked. And when we look at Watson and Crick's model with our A's, our T's, our C's, and G's, we find that the sequence of those nucleotides determines the outcome of our genes. The other thing that we already know is that there are 4 different DNA nucleotides. So one of the important questions we need to ask today is, how does 4 nucleotides, A, T, C, and G, code for all of the proteins that are in our body? And we know that proteins are made of building blocks known as amino acids. So the question really is, can we figure out the equation between 4 DNA nucleotides and the 20 different amino acids that we make in our body? So in order to talk about that, we're going to need some help. DNA is meant to be kept in the nucleus. It is the only copy of DNA we have in each cell. So we're going to need some helper molecules, and these are going to be RNA molecules. There are different types of RNA, and their use or their role in protein synthesis is going to be different depending on what type. Most importantly, here's one of your objectives. You need to know this chart back and forth. You need to be able to identify key differences in the structure of a DNA molecule. So one of the things with DNA versus RNA is we know that it's double-stranded, right? And that's how we get that double helix shape. RNA doesn't have 2 strands bonded together, it's just a single strand. You can see that they use different sugars as well. The other main difference is the bases. So A, T, C, and G, those are the bases that we find in the 4 nucleotides of DNA. In RNA, we don't have a T, we have a U instead. And so that's going to change up the base pairing rules a little bit because of that. As we know, DNA contains the original code of our genes. There's only one DNA molecule in each cell, and because of that, we have to protect it. So think of all the times that your cell needs to access the DNA in order to make more protein. Increasing the number of access points on your DNA molecule is going to increase the chance that the DNA gets exposed to something. So instead, the RNA contains the same information from the DNA, but in more of a disposable copy form. As we get into the 2 parts of protein synthesis, we'll be able to go into the specifics of the 2 main types of RNA that you need to know. But these are some key differences. So let's talk about breaking down protein synthesis. This page is the big picture page. So there are 2 parts. First is transcription. And when you transcribe something, that means to write across. So what are we writing across? We're writing the message of DNA into a sequence called mRNA. So it's a specific type of RNA, and we're going to get to that in a few slides. And basically, in the nucleus is where your DNA is and where it should stay. So when we need to access it to make proteins out in the cytoplasm, we're going to burn off a copy of just that gene sequence that we need, and we're going to make it out of RNA. And then that copy can leave the cell, not leave the cell, it can leave the nucleus and then go out into the cytoplasm where it can be translated, which is part B. And that's where we take the information that's in the mRNA and we convert it into a language that puts it into amino acids so that we can actually build our protein. So that's big picture, DNA to RNA to protein. Okay. Here's a little graphic here. It would be good if you labeled these parts. So DNA into the mRNA, That is transcription. Okay, that should be on your picture already. Then the mRNA that is produced from that is going to exit the nucleus, and it's going to come down here to the ribosome. And this part right here, all right, this is translation. So what's happening is the mRNA is being read by a ribosome. The ribosome is the gray structure right here. And the ribosome is reading the mRNA and using that to build these little chain of dots here, and each one of those is an amino acid. We see it arriving. And so that little chain of dots is going to be your polypeptide or your protein eventually that's going to grow. So very important to see, transcription happens in the nucleus. Translation happens in the cytoplasm at a ribosome. You need to know those two things. So transcription, what is it? All right, well, we're going to break apart our DNA molecule. We're going to split it in half so we can look at the bases on one side of it. And we're going to use the DNA as a template to build a strand of RNA. So we're going to need a helper. We're going to need an enzyme for this. RNA polymerase is going to be responsible for building the mRNA strands. You remember back from replication, we've seen this polymerase molecule, and it functions as DNA polymerase in DNA replication when it needs to build a DNA strand. But here, we are building RNA, so therefore it is an RNA polymerase molecule. And the whole point is we're going to take that DNA and make mRNA. So what does that M stand for? It means messenger, and the idea is we're going to send the message of DNA that's trapped in the nucleus out into the cytoplasm where it can be then translated. So we need to copy it. So if you think about it, the way I remember transcription, if you think in the olden days, back before they had photocopiers and printing presses and things like that, if you wanted an extra copy of a book, someone had to sit there and write all the words over again in a new book so that you could then take that copy elsewhere. That is what the mRNA does. And here's how it does it. Okay? We use the code from DNA, and it can match up base pair wise with a code in the RNA. So when we did DNA to DNA, it was easy. A to T, T to A, C to G, G to C. But now we've got some differences. Okay, if you look at the bottom here, G and C and G, they're going to be fine. And even a T in DNA is going to complementary base pair with A. But the problem is A in DNA would normally pair with thymine, but there is no thymine in RNA. So instead, it is going to base pair with U, which is called uracil. And when I say base pair, what I mean is we split the DNA molecule and we use one side of it to build a new RNA strand. So as it's building, it temporarily binds and pairs with that section of DNA, but it's just temporary. So you need to know this table. I'd put a big star on this page, and I would say make sure you know this pairing. So here's just a diagram to show you. The blue we see is double-stranded, right? That represents our DNA. We've pulled it apart, okay? This side of the DNA is not being transcribed at the moment. It is this side. And what's happening is the brown represents the mRNA that's being built. We see temporarily it is binding to the DNA nucleotides as it's matched, sequence for sequence. Okay. So just to zoom in on what we were just talking about there, here's our DNA molecule. And we've pulled apart, this is one strand of it, and then the growing RNA transcript, in other words, the new mRNA molecule that we are making, each sequence is matching up. Boom, boom, boom. We see T to A to U, C to G, and G to C. Okay, so now it's your turn. On your note handout, you'll see there is a DNA sequence. What I would like you to do is I'd like you to pause the video here and see if right above it you can write what the mRNA sequence would be, keeping in mind the base pairing rules. So pause the video. Okay, now that you've finished that, let's take a look at the answer. All right, so take a pause here, check to make sure you have all of your base pairs correct. Your G is going to be C in RNA, T in DNA will be A in RNA, A in DNA will be U in RNA, and a C in DNA will be G. And you go through the entire sequence that way. Okay? All right, so part 2, translation. Now that we have this mRNA sequence, okay, so we have the gene we want, we've copied it into mRNA, it leaves the nucleus and goes out into the cytoplasm. Now we need to convert it into the language of proteins. In other words, we need to go from a nucleotide sequence to an amino acid sequence. All right, and 3 key parts, there's the initiation, the elongation, and the termination. Luckily for you at the 9th grade level, you don't need to know all of these different pieces, but I will go into what you do need to know. So first of all, when we read the mRNA, one of the stumbling blocks, as I said before, how does ATCG, or in this case now AUCG, how does AUCG turn into 20 different amino acids? It's basically a math problem, and we did a lot of research into it, and what we found is that when you group the RNA nucleotides together and read them as a group of 3, that group of 3 will code for a specific amino acid. And so because of that, we call it a codon. Okay, right here. So the group of 3 nucleotides on the RNA strand is known as a codon. And each codon determines what amino acid will be added. So your amino acids are your building blocks of proteins. So if you look down this mRNA sequence here, we're going to group them together 1, 2, 3 at a time, and that's going to give us our first codon, whatever, that's going to code for 1 amino acid. Well, it would be really handy if we had some sort of matchup between, well, here are the 3 nucleotides, here are all the amino acids, what are they? Thanks to research, we now know this. So, get out your genetic code chart here, okay? And then what you're going to see is you have AGG. So let's assume this is a codon we've identified. I'm going to teach you how to read this code. Note that there are 3 different layers, 3 different levels of reading this code for RNA nucleotides. There's the center, then there's the next layer out, and then all the way on the outside. And then around the edges here, these are the names of the amino acids. So this is how you interpret a codon, okay? A is the first letter of our codon, okay? So right there, we can say that A being in the center circle, now all we have to look at are all of these possible amino acids. So all of these amino acids here have a codon that starts with the letter A. Then when we look at the second letter, which is G, now within the A quadrant, we're going to find the G in the next layer out. So here's AG. So that's narrowed us down to 2 possible amino acids. So we need to know that third letter in order to determine what the exact amino acid is. So A, G, G, and that's going to tell us that our amino acid here is arginine. Okay, so first round is the first letter, second is the second letter, and then the third ring is for your third letter. And when you look at all 3 of these bases, it's going to then point to what the amino acid is. It takes a little time to get used to this one, but let's do some practice. Okay, so here's the handout. You have it next to you, hopefully. Find out what the amino acids for these 3 codons. Okay, and then fill them in the blank space here. Pause the video, and then when you come back, we'll post the answers. Okay, so ACG codes for threonine, and that's how you say them, by the way. CUC codes for leucine, and UAA, all it says is stop. It doesn't say anything else. So that's obviously not the name of an amino acid, but rather the word stop, and we will get to what that means in just a second. Okay? So that's one thing you need to master, how to use a genetic code to take the codons of the mRNA and translate them into the amino acids. That's the basis of translation. But where do the amino acids come from? Are they just hanging around? Sort of. Okay, we're going to need a molecule that's going to bring our amino acid, like a taxi. So amino acids are just sort of floating free form out in the cytoplasm. They're just like hanging out, okay? And at the ribosome, okay, here's our ribosome, this gray thing. And then the green down here is our mRNA molecule. Let's say the ribosome is reading GGU, okay? GGU. When you look that up on your genetic code, you would find that it codes for the amino acid glycine. So now the ribosome is like, glycine, come on over. As soon as it reads that code, we're going to need a taxi service to bring our glycine molecule. So there you see the GLY. That's the glycine amino acid. It's going to hop a ride on this funky looking blue thing here. This is a tRNA molecule. It's made of RNA. And that is going to be brought here. So it's going to attach, and then it's going to transfer the amino acid to this growing amino acid chain that is already forming. So its job, the tRNA, is it transports amino acids from the cytoplasm to the ribosome. But one of the important things to know is that the tRNA is specific to a certain amino acid. So it's not like any tRNA could go out and pick up any amino acid. They match up with one another. And that's really more for security purposes. If we bring the wrong amino acid, then we build the wrong protein. It could have the wrong shape, which means it has the wrong function, mutation, bad things. Okay? So we want to make sure we get the right tRNA molecule with the right amino acid. And then that's going to come into the ribosome. It's going to say, look, I match. And then it's going to transfer the amino acid to the growing polypeptide, or the growing amino acid chain. So here's just another way that you could show tRNA. So you can see it's built of RNA molecules here. And the RNA molecule, it's twisted and folded into this weird shape. That's why they call it a T. If you notice, it sort of has this shape, T, okay, like a tRNA molecule. And then down here on this end, this is where the amino acid would attach. So our codon here is AUC. This is the mRNA. We're just showing a small piece of it. This is what's in the ribosome. So what is that going to ask for? That's going to be the code for isoleucine. So when the ribosome reads AUC, it calls out, I've got an AUC here. The genetic code tells us it's looking for isoleucine. So the tRNA molecule with the isoleucine is going to bring it into the ribosome and allow it to be attached to the growing polypeptide chain. Okay, that's great. How do you know where to start and where to end? We have to be told that. So there are special start codons and there are special stop codons. Start codon, there is only one. It is AUG. That sequence is present at the beginning of every gene of every living thing on this earth. Another point here is just to see how conserved that is through evolution. The idea that, yes, we are so different from one another, humans to bacteria to a tree to, an earthworm to, all sorts of living things. But yet, if you break down our DNA and you look at how we interpret the DNA to make our proteins, every single one of them starts with AUG. That's pretty powerful. So when the ribosome reads AUG, that's when it knows it's go time. We're going to be making a gene. And the way that I remember that this is the start codon, when does school start? It starts in August. So AUG is the code that is going to tell us when translation starts. It codes for a single amino acid that's called methionine. So that means every protein your body builds, the first amino acid in it is methionine. So once we start, everything goes on, and then the ribosome just keeps moving down the mRNA, reads a codon, calls for that amino acid to be brought in, and the chain keeps growing, link by link by link. How do we know when to stop? Well, we're going to need stop codons. There are actually 3 of them. We have UAG, UGA, and UAA. So if you look on your genetic code diagram, you'll see that for all 3 of those, the stop is what's attached to them. So instead of adding an amino acid, instead when the ribosome reads that codon, it says, oh, we're done? Okay. And then it releases the polypeptide chain, and then that can go. And we talked about how proteins fold into their final structure. So after that, the polypeptide chain is going to twist and fold into the final structure of whatever protein it may be. So that's sort of the theory behind translation. Now let's see if you can do translation. So here we have an mRNA sequence. All right. What I want you to do is, What I want you to do is I want you to use your genetic code, and I want you to determine what the amino acids are that would be coded by this sequence. If you notice, my mRNA strand here doesn't start with AUG. There's these extra C's. And that's sort of a leftover. It's really meant there to be sort of like a buffer. Like, you know, if the gene is the precious segment in the middle, you want to put extra garbage around the outside, sort of the same way that you would pack a vase in, you know, bubble wrap to protect it. So when the mRNA is moving through the cytoplasm, there are enzymes and all sorts of nasty things in the cytoplasm that could try and break this down, but we don't want it to affect this gene sequence. So whenever you are decoding, you need to find the AUG first. Okay, and you see that right here. Here's the AUG. From this point on is where you start grouping them in groups of 3. Okay, and what I've done here is I've color-coded it for you to make it a little bit easier. And notice on the end here we have something that doesn't add up and make 3. So this is not going to be a codon. This is not going to be a codon. It's just going to be junk. So pause the video here. See if you can translate this mRNA sequence into an amino acid sequence. Okay, and when we bring up our amino acid sequence, I just abbreviated them here. You can do that as well if you would like. When we look at the abbreviation, we have methionine, leucine, asparagine, arginine, cysteine, threonine, and then our stop codon. You always want to show that it ends with a stop codon. And that's how you transcribe and translate. So think about whether, if I gave you a sequence of DNA, could you show how it would be transcribed into mRNA? Could you then take that mRNA and translate it into amino acids? You will always be given a genetic code diagram. The genetic wheel here, you do not have to memorize anything on that. The only thing you need to know is that AUG is the start codon. That's what you need to look for. Okay, so with that genetic code wheel, could you then translate the mRNA into your amino acid sequence? And then that gives rise, that is the procedure for how we make all of the proteins in our body. So just to put translation together, you know, it's sort of hard to understand if you're trying to imagine things happening at once. I've got a little animation here to show you that can help. So, oh, that's your start codon and your stop. Sorry, forgot about that. So take a look at this animation, okay? This tRNA molecule brings methionine in. Here's the ribosome, the little hamburger-shaped thing, right? Right? Why did that guy bounce away? It was the wrong match, right? AGA codes for a different amino acid, okay? So as you see, the ribosome moves down, and then the ribosome is going to read the codon. The tRNA is going to bring in the amino acid, and then that blue thing that keeps coming in, that's just sealing in the amino acid sequence. We'll watch it again in a second. Hang on. Okay. So here we have the first methionine coming in. The ribosome forms around the mRNA, and now translation has begun. This one is wrong. See how it doesn't match? And so it gets bounced away. It won't be allowed to be added. This is the correct match. So this enzyme comes in and forms a bond between the amino acids. If you remember from chapter 2, that's a peptide bond. Ribosome moves down, next one comes in, amino acid is added to the growing chain. Okay? You don't need to know anything about this 5' cap or this 3'. But note we get down to the end, okay, and there we have our stop codon, and so it breaks off, and now this amino acid sequence can go and do its own thing, twisting and folding and forming a protein. Okay, so that's the end of transcription translation. We're going to do a number of review worksheets to make sure that you understand what the processes are, but also make sure vocabulary. There's a lot of vocab that goes with understanding this. In my opinion, wink, wink, nudge, nudge, if you could write a paragraph explaining all of the key players, meaning the molecules that are involved in transcription translation, and telling me where they occur and what they produce, you will be good to go on this section. So if you have any questions, please let me know. Make sure you refer to the objectives at the beginning of the OV.